Hello everyone, welcome again to Med Device Engineer X. And for today's episode, we're going to have a three-part series on the third part of servicing cardiovascular equipment, which is electrocardiography. And we're going to divide this section into three episode series, starting from cardiac physiology. The second part will be carrying out an ECG on what that clinicians actually do when they carry out an electrocardiogram. And the third one will be interpreting the ECG trace. Electrocardiography, well, let's start with what it is. An electrocardiography is essentially, it's short for ECG or EKG, and it is the recording of an electrical activity of the heart. What's the purpose of an ECG? An ECG is used to measure the heart's electrical conduction system. It detects the electrical impulses generated by the polarization and depolarization of cardiac tissue. And what it does, it translates this into a waveform, which is what we normally see in an ECG, which is an electrocardiogram. So in this series, we'll just learn the physiology of cardiac tissues before we actually dwell into the specifics of an ECG, before carrying out an ECG or interpreting the ECG trace. It's important to understand the chemical processes involved in initiating normal heart rhythms. In physiology, an action potential refers to a brief event where the electrical membrane potential of a cell rapidly rises and falls following a consistent trajectory. What are different functions in different cell types of an action potential? So in neurons, action potentials play a central role in cell-to-cell communication. In muscle cells, they initiate the chain of events leading to a muscle contraction. In the beta cells, specifically of the pancreas, action potentials trigger the release of insulin. So action potentials happen throughout various cells throughout the body and have different functions. And now, how does a generation of an action potential happen? Well, we're going to go more into detail with this with the later slides, but in general, how an action potential is generated, special voltage gated ion channels are normally located within the cell's plasma membrane, and these are responsible for generating action potentials. These channels, they remain close near the cell's resting potential but rapidly open when the membrane potential reaches a specific threshold. This is called the threshold voltage, as you can see from this diagram. Upon opening, they allow an inward flow of sodium ions, which leads to a depolarization, as we see in number 2. So sodium ions come in, and then this process continuously explodes until all available ion channels are open, resulting in an increase in membrane potential. Once it reaches its peak around here, the polarity of the plasma brain reverses. As you can see from the diagram, it goes down again, and the ion channels rapidly inactivate. So sodium ions are then actively transported out of the membrane, while potassium channels are activated, causing an outward current of potassium ions and returning the membrane to its resting state, which is around plus, I believe it's 70 millivolts. Following an action potential, there is a transient negative shift known as the after hypolarization or refractory period, which prevents the action potential from traveling back along the membrane. Now, there's different types of action potentials that exist in animal cells. Those generated by voltage-gated sodium channels and those by voltage-gated calcium channels. Sodium-based channels and sodium-based action potentials typically last for less than 1 millisecond only, while calcium-based ones may last for 100 milliseconds or longer. In some neurons, slow calcium spikes drive a long burst of rapidly emitted sodium spikes. In cardiac muscles, a fast sodium spikes primes the rapid onset of a calcium spike, leading to muscle contraction. So what's the difference between action potential and a cardio action potential? In simple terms, an action potential refers to a brief electrical event that happens in various types of cells, as we've discussed before, so in the beta cells of the pancreas, in the neurons, in muscle cells, which allows them to communicate and contract. Whereas in cardio action potential, this specifically only happens in the cells of the heart, as the name says, cardiac. It's similar in the sense of a short burst of electrical activity, but it's only unique to the heart cells and is essential for coordinating the heart's rhythm and pumping action. So while both involve electrical signals, a cardiac action potential is specific to the heart's cells, whereas an action potential can happen in different types of cells throughout the body. And the diagram, as you can see in the slide, is different phases of a cardiac action potential. And in the next episode, we'll discuss in detail, starting from phase 4, 0, 1, 2, 3, what happens in each phase of the cardiac action potential. Thank you for watching, and I'll see you in the next episode.